Right. All good? All right. So thanks everyone for coming on over to the Brain and Mind Center. So I was asked by Fernando Calamonte to do a sort of bit of an introduction to neuroanatomy for people that aren't necessarily steeped in the sort of tradition of sitting and looking at cadavers and walking through in excruciating detail all the little tiny structures and trying to remember exactly what this Latin word means and that Latin word doesn't mean. And I eagerly agreed because I'm a bit of a neuroanatomy geek. I really like thinking about functional neuroanatomy and how the system fits together. I'm convinced there are simple rules. I haven't found them yet, but I'm convinced there are simple rules under the surface. But what I hadn't realized was just how dry of a topic this is and how essentially you've got to really think about how to carve up the system in a way that's hopefully interesting to you, and not too glossy, but also not too detailed that you basically fall asleep. So I'm going to do my best at that. One important thing at the front, I really, really want you to interrupt. We've got a whiteboard there. I can draw things. If there's a part of it that doesn't make sense to you, please just let me know. We're going to spread this out over 3 weeks. They're not contiguous because next week there's a forefront conference, but today I'm going to start off talking about gross anatomy, which is kind of just like the high level systems, how they're, what's the hippocampus, what's the basal ganglia, what are the cerebellar cortices. But there's no reason that if we don't get to the end of it, I couldn't just fold this over into the next one, right? They're not partitioned in any really robust order. So what I'm going to try to do in the first one is give you this like sort of bird's eye view of neuroanatomy, all the different parts, kind of a vague way to think about them that's helpful. In the second part, which is going to be in 2 weeks, I'm going to talk about the microcircuitry in a bit more detail. So try to think about how inhibitory and excitatory populations coordinate their activity to give rise to sort of system dynamics in these different circuits. That's going to get pretty gnarly, and we'll do our best to sort of get through that without too many people falling asleep. And then in the last one, which will come in 4 weeks' time, or 3 weeks' time, sorry, I'm going to try and give you a bit of an idea about how I think they all fit together. So this last one's going to be really a sort of functional neuroanatomy. The rest of them are going to be trying to just walk through, what it looks like, how they're wired up together, and then how they all might come together to work as a single organ. The main things that I'm going to be talking about, obviously the cerebral cortex. This is the sort of big lumpy bit on the outside that many people mistake for the whole brain. We'll talk about that quite a bit. Things get labeled into this second category, what we call the subcortex, which is quite insulting to the poor subcortical structures. They've been around for far longer than big developed cortex. But this includes regions like the thalamus, which are a very, very important central processing hub in the brain, the basal ganglia, which people may know of through their dysfunction in disorders like Huntington's and Parkinson's disease, the cerebellum, which is also called the little brain, but actually has over 60% of the neurons in the entire central nervous system, and it's performing really fascinating roles in whole brain dynamics. The hippocampus, which is quite infamous for being a center for episodic memory, your ability to recall, and it becomes dysfunctional in disorders like Alzheimer's disease. People will have heard of the amygdala, the superior and inferior colliculi, and the list goes on and on and on. And then sort of put down the bottom here of the list, even though I think these are some of the most important bits of the brain stem and the spinal cord, thinking about how the brain coordinates its activity with the rest of the body and hence the world. Okay, and as I said, please ask questions. So just raise your hand. I'm more than happy to kind of dive in. I can go back. I can try to elaborate on things or give you another way of thinking about some of these concepts if it's helpful. So I just wanted to start out very quickly with the background check. I think I know a fair few people in the audience. So there's a few physicists here, people from psychology. Can I just get a feel for where other people are from? So how many people have like a science background? Yeah. Okay, so we've got mainly scientists, that's good. How many people feel like they know everything there is to know about neuroanatomy? Good, it was a trick question. It's impossible to know everything. Oh, hey Joe, what's up? We'll just wait for them to trickle in. Bloody engineers. So, all right. Connor, don't sit behind the pole. Okay, so I think we've got a fairly diverse audience. Please stop me if I'm going too slow or too fast as well. I didn't really know how to pitch it, so I've just tried to do my best bird's eye view. So, starting point, gross anatomy. I thought I'd put this up because it's not a funny joke, whatever. Okay, all right. So, starting point, we need to get some terminology down. Neuroanatomy has all these funny words about where you're looking and where things are with respect to everything else. If you think about this, it's actually really important if you're an anatomist. You're looking at a slide, you've taken a cut through the brain in some axis, you want to know what that axis is, and you'd like to know which end is front, back, left or right, up or down. Now, the funny words we use is because we have different reference points that have some meaning to us. So you can imagine that one way to cut the brain up is to do this kind of back to front. Here we're looking at a medial view of the brain, cerebellum, brainstem, thalamus. This here is the callosum, this big white matter bundle that connects between the two cortical hemispheres, and this big lumpy bit on the outside is the cerebral cortex. So you can imagine starting at the back, the occipital pole, and going forward to the frontal pole. Or you could just as easily imagine starting in the spinal cord, which would trickle down here through the vertebral column and coming up to the top of the brain. And so it's a little bit ambiguous as to which way is up or down or front or back. And so we have a set of words that we use. Up and down in actual space is inferior and superior, and front and back is posterior and anterior. But one thing you'll see a lot in anatomical language is caudal and rostral, and ventral and dorsal. So thinking about this in terms of humans is a little tricky because we no longer have a tail. Our nose isn't particularly impressive. We don't have a dorsal fin, for example. We're not a snake. We don't have all of our exit and entry points on our ventral surface necessarily. Some of them wrap around. So a way to think about this is rostral is like your rostrum, your nose, and caudal is like your tail. That's another name for tail is the caudal pole. So that's a caudal region and a rostral region. So if I was in the thalamus and I said, what's rostral, it'd be in this direction. Or if I was in the motor cortex and I wanted to go to the frontal pole, I'd be going in a rostral direction. As I went to the occipital lobe, I'd be going in a caudal direction. And then dorsal and ventral, I think of the dorsal fin and the ventral just being the front where the vents are. Another thing you might see a little bit, particularly if you do neuroimaging, is these terms sagittal, coronal, and either transverse or axial plane. And I think it's worth just having a little Google anytime you come up against these to make sure that you know which plane you're in. There are some sort of simple heuristics. So an axial is going from left to right, like someone putting on Axe body spray. A coronal is like putting a crown on someone's head. So if I was putting a crown on someone's head in front of me, I would chop right through the middle of their brain like that. And a sagittal plane is from Sagittarius, which is the archer. So it's cutting in this direction. So you could just, you can come up with heuristics, or you can just bloody Google sagittal and it'll tell you what it is. All right, some other terms that I think are important. I remember back in med school, anytime someone showed me these fancy terms, I would immediately fall asleep. So I'm not going to belabor it, but I want you to be aware of these terms in case they come up. It turns out that when we're developing in embryo, we actually don't look very much like this kind of brain on the other side, this other side with all the lumps and specialization. We essentially look like a little tube. And the tube begins out of a little clump of cells that then differentiate, have a little hollow between them. And then you start to get these little pouches that form that people will call the hindbrain, the midbrain, and the forebrain. Now, this is really interesting for people that care about things like genetic disorders because there's certain genetic rules that tell you whether or not you end up being a forebrain cell or a midbrain cell. And then if you extrapolate forward as the tube starts to differentiate even further, you start to get these terms like myelencephalon, medencephalon, mesencephalon. These basically all end up becoming the brainstem, which is this little structure here. The diencephalon, which is the thalamus, this central processing station, and the hypothalamus. Even though these guys aren't sitting on the top of the hierarchy, they tend to be some of the most important brain structures for defining the function of the circuit. And then on the end, you've got this telencephalon, which has the cerebral hemispheres, the basal ganglia, the amygdala, the hippocampus. So most of the work that people do, in fact, if you asked someone doing neuroimaging what they cared about, most people were just hanging out up in this little pole here. They don't do a whole lot of work down in this area. One of the things I'm going to argue throughout this lecture series is that thinking about these other structures and constraints are incredibly important if you want to understand what it is that the cerebral cortex can do. The second thing that I wanted to mention, and this will come out a little bit in some of the later talks, the cerebral cortex is actually just like a sort of out bulging from this diencephalon, part of the hypothalamus. And so we'll talk about this a little bit later, but it looks like, if you imagine this is like one of those, you guys seen those like party balloons that are all sort of wrinkly and stuff, and then as you blow them out, they have different shapes to them. You could think of like this is this little tube that has these little kinks in it. And if I blew this up, what happens in humans is that this part of the tube becomes these really big, lumpy cerebral cortices, right, and basal ganglia and amygdala and hippocampus. But every vertebrate has exactly the same bowel plan, the same architecture. So some of them have smaller cerebral cortices relative to the size of their brainstem and thalamus. But you don't have, one of the old ideas of how the brain was organized is that you started out with the brainstem, right? They call this the reptile brain. And all you've got is the brainstem and you just function with that. And then as you develop into a mammal over evolutionary time, then all of a sudden you get this thalamus and hypothalamus. And then only in the beautiful humans that were, you know, the clouds parted and we were put down on Earth to destroy it and vote for Donald Trump, have this cerebral cortex on top. And that's just not the right way to think about it. There's this neural tube that gets ballooned out in different proportions in different species, and that will give them different behavioral capacities. So this is just another way to show you this same point. We start out as a little green tube. We develop bulges in the tube as if we're blowing it up. They start to differentiate with different genetic signals. Interesting things start to happen, like the cerebella starts to pouch out from this little purple zone. The thalamus starts to form in this orange zone. This green bit's called our tectum. We were just chatting about the superior colliculus before, and your ability to move your eyes around the world all depends on things in this zone. And basically everything up here in red is what ends up becoming telencephalon, and hence the cortex and basal ganglia. But it really grows out of this same bulge here, which ends up being your hypothalamus, which is really important for setting the kinds of things that you're sort of trying to achieve in the world. So this is a slightly more complicated diagram just to show you that this is like an oversimplification. So this is that same diagram down the bottom, but just sort of blown up into slightly more detail. The big difference that this, or the point I'm trying to convey with this diagram, is that there's a lot of connections that are really well defined across all of these different locations. So you can imagine the eye here is going to have a particular input that's going to come in, synapse with the thalamus and the tectum. This is the superior colliculus I was talking about. The colliculus will come back and hit the thalamus, which is then going to send back up into the cortex. And so one of the things we'll cover predominantly next week, or the second lecture, is how those different connections shape the function of these different substructures in the brain. But we're left with a bit of a problem here, because this diagram I'm showing you looks absolutely nothing like the kind of thing you would see if you Googled the word brain, right? This is what a human brain looks like, colored up. So my goal with this whole thing, this whole set of lectures, is to be able to tell stories or try to get you to link the structures that would happen as we're developing to these things that you're seeing in a whole mature brain. All right, any questions before we dive in? All right, so the human brain. Actually, this outside bit here, what people would typically call the brain, is really just the cortex, the cerebral cortex. It's quite lumpy. You can see all these grooves and sort of bulges throughout it. It's a bit of it actually doesn't look anything like that when you look under the hood. It's actually a really thin strip. It almost looks like the thickness of about 2 or 3 playing cards spread out over the size of a tennis court, but then it gets pushed and squished down into a skull. And so a really effective way to push and squish something as big as a tennis court down into a skull is to wrap it around and convolute it, and basically increase your ability to have a high surface area in a small volume. So this lumpiness of the cortex is really due to a lot of the physical constraints imposed by our skulls. There are typically described 4 main lobes of the cerebral cortex, although there's a couple extra little added mini lobes that I'll talk about in a second. So this is looking at the cerebral cortex from the left-hand side. You can see here there's a temporal lobe, occipital lobe, parietal lobe. All of these are kind of split up by these main, what we call sulci in the brain, and then the frontal lobe at the front, which people spend a lot of time thinking about. So just to give you a bit of a bird's eye view here, so the occipital lobe is the primary destination for a lot of the inputs from the retina, from our eye. So they go through the thalamus, to the lateral geniculate nucleus, and send this huge projection into V1, which is the area that is right at the back of the occipital cortex. And then from there, there's lots of connections within the cortex that then process the information either to identify which object you're looking at or with how you might interact with that object on this stream up towards the parietal lobe. So imagine we're going down the object recognition pathway down to the temporal lobe. So down in the temporal lobe, let's forget the top bit for a second. This is just like a little diagram from a neat paper that tried to mimic the processing pathway of the visual cortex using a set of recurrent neural nets. The idea is that as the input comes in from the retina, it hits the thalamus, goes to the cortex, and then it's processed at increasing levels of abstraction as you move up this occipitotemporal gradient. So here I might be differentiating between lines that are vertical versus horizontal, and as I move out, I start to recognize shapes and distinguish them. And by the time I'm out into the more mature areas in the ventral temporal cortex, I'm recognizing different faces from one another. This is the general idea. The temporal lobe also houses the hippocampus, which is sort of deep to the projection we're looking at here, which is a major seat of memory. And I'll talk about how those two relate together in a second. And also up here is the primary auditory cortex, which receives the input from the cochlea through your ears and is processing in much the same way as the temporal, the visual flow is processing both where something was, but also what the content of that signal was. This is a famous picture from a paper back in 1991, Fellman and Van Essen, where they mapped I believe it was a macaque cortex, and they were trying to work out the number of connections between all the different areas. And the take home is supposed to be that it's a hot mess in there, right? There's tons and tons of different connections. In fact, if you calculate the number of inputs that come from the lateral geniculate nucleus, which is the area that receives the input from the retina, the proportion of those to the connections that come back to V1, this primary visual area, it's about 90 or 95% of the inputs are coming from cortex, and about 5 are coming from the retina. So we know that those 5 are really important functionally, but we also know that the balance of power is in favor of the cortex. It can actually bias this system to whatever patterns it has within it. All right, the parietal lobes, so they're sitting up more superior to the occipital lobes. When the occipital lobes get their information in, they process some of it down that object recognition pathway, and a number of the other connections, particularly related to movement, will get pushed up into this parietal lobe. And I like to think about this in the context of something called affordances, which is that the parietal lobe is telling you how you can interact with the world. If you see a chair and you want to sit down, the parietal lobe is telling you how to sit down in the chair. Whereas if you see the chair and you're angry, it's telling you how you can pick it up and throw it at someone. Right? So it's telling you the different ways that you could interact with the structure of the world as you see it, both from the visual stream, but also from the sensory stream. So sitting up here in what's called the sensory cortices, the sort of most rostral section in the parietal lobe, are the inputs that come through your spinal cord, from your skin and your body, that tell you where your body is in space. So another way to think about the parietal lobe, because you can also imagine here the auditory cortex, is they're all feeding their sensory representations or inputs, guesses about the world. And there's this multimodal integration where you're saying, how does the auditory signal relate to the visual signal, relate to the touch signal that I'm seeing? And so this lobe is typically classified as what we call an association cortex, because it's doing this kind of binding between these different modalities and trying to find this sort of more low-dimensional structure in all of these really high-dimensional sensory systems. All right, and then up the front in pink, we have the frontal lobe. It's very hard to find GIFs and pictures to describe all the things the frontal lobe can do. So right here at the most caudal area in the frontal lobe is what we call the motor strip or the motor cortex. So different parts of this will move different parts of your body. So out here, this might move your lips, and here this might move your hands or fingers, and on the other side of the frontal cortex, it might move your legs. We'll talk about that a little bit more in the second lecture about how that's wired up. As you move more rostral, you start to become more abstract, and you start to coordinate combinations of these different muscle groups. So let's say that I wanted to pick up a ball, throw it in the air, and then kick it. It's going to be some area out here that's going to have some combination of signals that will coordinate those activities in this motor strip to allow me to perform that function. And as we move even more rostral to the rostral pole, the idea is we're becoming more and more abstract. So let's say I wanted to make a plan about kicking the ball, that might be here. But if I wanted to make a plan to kick the ball when I get home with my kids, maybe that's further here. And if I want to make a plan to kick a ball when I'm traveling overseas in 3 years to go visit a friend to kick a ball, it might be out here. So the idea is that you're moving more abstractly or more protracted in time as you move away from this really embodied motor cortex out into this frontal pole. There's also some other fascinating things in the frontal cortex. I'm not going to talk about all of them, but one of them is quite interesting. There's this area, the right inferior frontal gyrus and the left inferior frontal gyrus, that are really important for ceasing behavior. So if you're in the middle, if you're driving down the street and then the light turns red, it'll help you slam on the brakes. This forms the basis of the cortical node and what we call the hyperdirect pathway, and then I'll come up when we talk about the basal ganglia. And there's a bunch of other stuff going on there too, but this is just a sort of bit of a high level. All right, so I talked about the sulci and gyri. This is just kind of like a cartoon to hammer this home. The idea is that if you're trying to fit everything into the brain, you need to fold it up. So we call that the lumpy bits the gyrus or the gyri, and the little pits the sulci or a sulcus. And originally, when people were mapping out the cortex, they noticed that there's these consistencies. So this here, this central gyrus, actually is consistent in every person. It has the motor cortex in front and the sensory cortex behind it. There's also the superior temporal gyrus sitting here, where it, which is next to the superior temporal sulcus and houses the auditory cortex. And you can go through and you can kind of split the brain up with these different sulci and gyri. And so it might be that if you're doing an imaging experiment, you don't care so much about whether or not it's in the right inferior frontal gyrus. What you want to know is it in the middle frontal gyrus or the superior frontal gyrus? And this is just another way that you can describe the system. This has fallen out of favor a little bit with modern neuroimaging techniques because we have a sort of agreed upon coordinate system. And so we instead use that coordinate system, and then you can go and have yourself a good old time using whatever label you'd like to that coordinate system. All right, so I mentioned that there were a couple of sort of mini lobes or things that don't really fit into the 4-lobe story. One that's quite important is called the cingulate cortex. And it's called the cingulate cortex because it wraps around the cingulum bundle. I said before, this is a sort of white matter highway that connects the left and the right hemisphere together. This is quite an old cortex in that, like pretty much every mammal has a really well-developed cingulate cortex. And it doesn't really fit neatly into any of the categories that I suggested before. And part of that is that it crosses a lot of these boundaries. So here, this part of the cingulate cortex is going to be sort of mirroring and mapping some of these orbitofrontal functions, which receive all these inputs from the hypothalamus down here and the midbrain. They're sort of dousing it in motivational chemicals that tell you whether you should care about something now or not. Whereas as you move up, this dorsal anterior cingulate region is sort of doing something kind of like what the more superior parts of the frontal cortex do, and it gets a lot of inputs in from the amygdala and the noradrenergic system. And as I move further back, I'm going to hit what we call the motor cingulate, the sensory cingulate, the paracingulate, the retrosplenial cortex. And so you can sort of think of the cingulate cortex as sort of like a little mini microcosm of whatever cortical region is near it. Now that's not a perfect way to think about it, but it's certainly a sort of nice starting point. Just to show you that these things are kind of mapped out in the anatomy, if you care deeply about this area of the brain, there's a really lovely paper in 2007 where what they did is they put little seeds in the brain from the caudal to the rostral dimension, both in a superior and inferior strip, and then mapped their what's called resting state connectivity. And you can see that if you start at the back of the cingulate, you've got a really local hotspot. But then as you start to track forward, you can see more and more of that anterior connectivity popping into the system. So they're really sort of mirroring almost like a searchlight, whichever region of cortex is sitting directly near them. But it's not that simple because there's areas of anti-correlation and distributed correlation around the brain. So if you're interested, that's a good starting point. The second and other sort of mini lobe that doesn't quite fit, we call the insula. And so that picture I showed you before that looks like the brain on the side, if you opened it out with the scalpels here, or just the protractors, you'll see that there's another area of cortex. You can see the insula is not a particularly clever name, it's just inside. But it's a really interesting area. It has connections from a lot of internal autonomic structures and also has a lot of inputs from the amygdala. And so we'll talk about that a little bit later about its function. This is just a little diagram that's far too busy, but just to show you that you're getting inputs to the insula come from all around the brain. So they're coming from the vestibular system, which controls your balance, the spinal lamina, this is sensory, the solitary tract, which is coming through the vagus that collects all of the autonomic information from your gut, out to the frontal cortex, and then olfactory inputs in areas like the amygdala. So the insula is kind of like a microcosm for the whole brain, kind of like the cingulate as well. I thought I'd throw this in there just in case this is the kind of frame of reference people had. So this is what we would call resting state networks. This was a seminal study back in 2011, Tom Yo, Randy Buckner's group, where they took, I think at the time it was 500 or 1,000 people that underwent resting state scanning, which is where you lie in a scanner collecting functional data, but you're not told to do anything in particular. And what they were trying to do was look for consistent patterns of activity which were highly correlated with each other linearly. And they used some fancy machine learning to collapse this down. But essentially what they found is these solutions that either have 7 networks or 17 networks, that the idea is that what they're doing is they're trying to find these correlations between these little areas of the brain that are distinct. So they find the heat in these little modules and they call that a network. And so you can debate until the cows come home whether this is a terrible name for it, but it is, for better or for worse, the name that we use in the field. And so the idea is that this isn't now an anatomical parcellation, it's a functional parcellation in the sense that we've taken some data and we've asked how the data was organized. And so you can see that some of the networks respect anatomical boundaries. Here, the motor and sensory cortices have very similar time series in a resting state study, whereas other ones have these really interesting patterns where, say, some of the frontal lobe here, the medial frontal gyrus, is connected into parts of the posterior parietal cortex and parts of the middle temporal gyrus and the anterior insula. And so what we, for better or for worse, as I said, we call these resting state networks. There's lots of different solutions for them. This is the 7 and the 17. If you're interested in these, you can go and look at the names of them, but just to sort of know that's, they're not related to the neuroanatomy per se, they are composed of functional data. Another thing that's quite popular nowadays is to essentially not parcellate the brain. So here what they've done is they've done some kind of connectivity matrix and they've then gone through and with some rule chopped it up into different squares that they say form what they call a consistent network. And you can do this in data-driven ways or model-driven ways. There's lots of different ways to do it. Another strategy is to essentially not attempt to cluster the data, but instead perform something like a dimensionality reduction on the data. So here they use something like non-negative matrix factorization to take all of the different spots in the brain and cluster it up. And you can see that there's these basic gradients where at one end you've got these kind of frontoparietal temporal areas, and then they're anti-correlated or separable from this big sensory motor insular strip and this area of V1. And so then people will say, okay, then maybe this organization is a better way to think about the actual pattern of activity in the brain, because this is what the brain actually looks like when you're letting it sit in a scanner. This harkens back to an idea from the late 90s by Marcel Messelem called From Sensation to Cognition, which, if you're curious, is probably one of the better papers I've read, where he basically argued this graph about 30 years before people found it from first principles, which is pretty impressive. So a cool thing about these gradients is that they reflect functionality as well. So a lot of what I've been talking about today is just putting things into bins, but it turns out that if you do data-driven analyses, you can sometimes just recover those functions. So in this case, the first gradient that Margulies found, so if we just pull these blue and green together, relate to things like motor movements, eye movements, visual perception, pain, action. And if we go to the other end of the gradient, where the areas are red, now we're looking at things like social cognition, emotion, verbal semantics, autobiographical memory. And so the idea is that we might be able to come up with these data-driven ontologies where we can try to sort of orient the functional, the different behaviors that we are capable of along these different axes. So a way to think about these is based on a really interesting pattern that's developed over evolutionary time. This is another beautiful paper by Randy Buckner and Fenner-Krenen. Sorry, it's cutting off on the edge, but it's 2013 in Trends in Cognitive Sciences. And The argument that Randy and Fenner were making was that if you look at the kinds of structures in the cortex that are preserved over evolutionary time, you can track these basic motor, sensory, this would be auditory, these would be internal like visceromotor signals, and then visual, back all the way down the cladistics tree, and you can see those exact same patterns recurring in all these different examples. So every animal we know of has, a motor strip so they can move, they have a visual input, they have auditory input. Some of them have more visual architecture than others. A bat doesn't have a whole lot of visual. It's got, it can move, and it has a really nice advanced auditory cortex, so it can do its little sonar blips. But the concept here, which I think is quite beautiful, is that what's happened over time is that we've moved up, we've basically expanded out this little zone that's interacting between these different inputs that are, say, coming from the world, from the, this is, say the sensory inputs and the visual inputs. What we've done is we've expanded out these areas that allow the communication and the sort of differential connection of particular inputs in these different categories. And so the way that they frame this is they say that these red and blue signals are tethered to the world. They're tethered to the types of inputs you're getting from your retina and, say, from your skin. But these white regions are untethered. And so one characteristic of humans is that we're relatively untethered to the inputs that we get, and we can combine them in lots of interesting ways. I think this is a really big part of the story if we're trying to work out what it is that is a little bit different about humans compared to other animals that we look at. Another really interesting thing that happens across the cortex is a change in timescale. So Chris Honey, Uri Hassan, Chris Baldessano have done some really cool work. This is just one example where people listened to a narrative structure in a scanner, so an fMRI scanner, and they were trying to map what happened when they played them sounds, the narrative structure backwards. So they're getting lots of sound input but doesn't have any meaning to it, versus words, versus slightly more complicated sentences, and then to the whole level of the narrative. And what you can see is that the sensory system is really tapped into just getting perturbed in any way, shape, or form, irrespective of whether it has structure. But then the temporoparietal junction, these frontal cortices, are tapped into these much more long time scale and higher level, more high dimensional structures in the input stream. So I think this is actually a really fundamental result. And they've shown similar things for the visual stream as well. I'm sure it would be the case for the sensory stream, though it's a little harder to imagine how you would do that experiment. I think this is a really important phenomena, that you can imagine these tethered structures tapping into these really high frequency but quick refresh rate signals, and then these untethered structures basically trying to find these much more refined abstract patterns between the input streams. So I thought some of this was, a little bit of an onslaught of detail. So I try to put into a bit of a context for you. And one of my favorite things that I've learned in my short time in neuroscience is just how wrong the medical textbooks are about visual perception. So when I was in medical school, we got taught about visual perception by just looking at the eyeball, right? It's where the light comes in, there's a retina there, the retina passes the information to the cortex, and that's pretty much the whole game, right? All I gotta do is point my retina at something and I can see it. So really the analogy of this is that our retina is like a camera. We're pointing the camera around the world, we see something, and then we move on. But as I said before, there's actually way more connectivity coming back from the cortex down to lower layers of cortex, which can then shape what's going on. And so a better analogy is actually that perception's more like active. It's more like something we do than something that happens to us. So I've got this, if you guys don't know the show Review, it's absolutely hilarious. But what he's doing here is he's blind and he's feeling out the object. And so even though it doesn't seem like it, our world seems stable to us. You actually look at your eyeballs, if you have someone looking around a room, they're constantly saccading, and they're making these little hypotheses about the world, and they're collecting evidence about those hypotheses and moving on. So it actually is a lot more like touching something you can't see. It just feels like we're watching a movie that's quite static. So a way to think about this is that instead of seeing the world as it is, we're actually seeing the world as you expect it to be often. And so there's this constant cycling between these two. If I expect something to be there, if I expect to see a friend walk through the door, and they actually walk through the door, I'll have a much quicker reaction time to responding to to them walking through. So people would say that this is sort of describing perception as an active process rather than a passive process. So this gets really interesting because then you can imagine seeing the world as it actually is versus being creative and imagining what you would like to see. So this is, like a little ice cream shaped like the world. And the cool thing is that there's a lot of nice evidence now that this level of switching between seeing the world in a veridical sense and imagining the world relates to this kind of mesoscale architecture of these levels of networks we talked about. So I didn't belabor the point here, but this here is a representation of what we call the default mode network. This is what we call the ventral attention network, and this is what we call the dorsal attention network. And so you can think about the ventral attention network as kind of arbitrating between whether you focus on the world or you focus on some imagination of the world as you would like it to be. So to give you this in a slightly more sort of intuitive framework, imagine by this point we've been talking for 40 minutes, you're really bored, you're kind of starting to think about how nice it would be to be outside in this beautiful weather, but then the beauty of the slides I'm presenting catches your attention. What that does is it recruits your ventral attention network to inhibit your mind wandering, and then you recruit your dorsal attention network to pay attention to the task again. So you can imagine that your brain is constantly shifting back and forth between these different modes of processing. All right, so let me just take a drink. So in the last 15 to 20 minutes or so, I'm just going to whip through a few high levels of the rest of the brain. And this is really to set us up for the talk in a couple of weeks' time. So first off, the spinal cord. So here I'm showing the brainstem, but recall the brainstem is a continuation of the spinal cord which runs down your vertebral column. And so this is a sort of a nice graphic showing you what that might look like. Here we've got this brain on top of the brainstem and the thalamus. And if we follow that all the way down, there's these different segments of what we would call the cervical spinal cord. And the cervical means neck in Latin. So these are receiving sensory input and controlling the muscles of the neck through these different segments. If you ever see these different things, see 1, 2, 3, 4, 5, 6, 7, 8, what they're telling you about is the vertebral level. So there's a level here of all the different vertebral bones that are sort of stacked on top of each other, like a set of Lego blocks that are a little bit more flexible. And every time you stack one on top of another, there's a little gap. And a little nerve root leaves that gap or comes in through that gap. And so clinicians care about this because that gap can get closed by a bulging disc or some kind of a trauma. And so we'd like to be able to tell you which area that the lesion happened. This is called the thoracic spine. Thoracic means body, I think. I can't remember exactly what it means, but it represents body. These are going to be the nerve roots that are responding to the input of your trunk muscles, so your abs and your pectoral muscles, and also down into your abdomen. The lumbar spine is down into your legs, so hips, your front of your legs, quadriceps, and down to the feet. And then the sacral and the coccygeal are your genitourinary function. So just to sort of orient you, there's this kind of structure running all the way down. And Fernando and I were talking about this cauda equina at the bottom, these nerve roots that kind of fan out at the bottom. If we took a cut here through an axial cut through an axial cut through the spinal cord, it would look something like this. This is going to be the top, so the back here, the posterior portion, and this is going to be the anterior portion. You'll see these two different kind of structures within. The little thing that looks like an H is called the gray matter, and the white part on the outside is the white matter. White matter is the myelination, the fat that's insulating the fibers that are running up and down the spinal cord from the body to the brain and back and forth. And the gray is the cell bodies or the unmyelinated fibers that are coordinating that activity or allowing the interconnection. I don't want to go into a huge amount of detail here, but you'll notice that there's a bunch of different tracts that are running up and down the spinal cord. These ones here are quite important. These are what's called the dorsal column tract. So they're taking sensory input from your legs and your arms and running it up into the brain. And then I think, which one is it here? Yeah, this is the lateral corticospinal tract. So the motor tract here is running all the way down. Let's say you wanted to move your right leg. It would come out one of these foramina. It would hit the spinal cord, come out the foramina, and then it would extend the muscle to make you activate that movement. An important point, originally people took this logic of this spinal cord and thought, this must just go all the way up into the brain. And you just then basically just like draw a line. All you need to do is draw the line all the way through the brain, and you've got the motor bits and the sensory bits. Because you notice that all the sensory bits are kind of here and laterally, whereas the motor bits are kind of medial and ventral. So this is what it might look like in that original model I showed you. You might have a dotted line separating the motor from the sensory that would went all the way up into the cerebral cortex. It turns out that's actually incorrect. If you go back and look at the developmental trajectory, you can actually see what happens is it bends around, and then it actually takes a sort of U-turn. And so the sensory stuff is all in this sort of what's called the alar border, the basal border, sorry. And then the alar border has all of the motor movements. So this actually is sort of confirmatory evidence for this idea that the main thing you need your cortex for is planning motor actions and sequences. All right, just north of the spinal cord is the brainstem. There are kind of three main structures in the brainstem. So here, showing the brainstem zoomed in. The most inferior portion, we have the medulla. The medulla has a bunch of inputs from things like the visceral motor organs, the gut, but also areas like the vestibular system, which basically keeps you balanced and fails completely when you've had a few drinks. Just above that is the pons. The pons is big and bulgy, particularly in humans. It has all these little association nuclei, which I think are super interesting, and they'll come up in the next couple talks. And it's the major input into the cerebellum, but it also has a number of structures that come up and arouse activity in the thalamus as well as the rest of the cortex, and what we call the ascending arousal system. The midbrain is a relatively small area. It also has some more of those arousal structures and some particularly important motor nuclei for controlling eye movements. All these little brands of spaghetti that you can see sticking off here are what we call the cranial nerve roots. Clinicians get really worried about this because let's say you had a stroke in part of your pons, we can try to track where that may have occurred by which of the different nerves we can tell are actually impaired on a clinical examination. This is now less important that we have good non-invasive imaging, but this was a sort of tradition in clinical science. Another thing that's running through the brainstem, and the brainstem is coordinating, is what we call the autonomic nervous system. So the autonomic nervous system is kind of controlling your fight and flight and your rest and digest. So your ability to kind of automatically keep everything in homeostasis. And so the way it kind of works is that there's these nuclei within your spinal cord that have these little roots that come out, hit these ganglia, which then go out and hit the end organs. So they might hit, the genitourinary system or the kidneys or the liver up into the brain. It's not shown in this diagram, but the brainstem has control over this system, and it can kind of turn the volume up and down on whether or not you're pushing into one or the other mode. That'll become relevant later. I included this diagram just because I think it's super cool. Another thing to remember is that the brainstem has a bunch of what we call central pattern generators. So these are kind of like hidden nodes in the neural network that have these connections into these effectors that you can imagine running through the spinal cord. And the idea is that if we tweak just one of these central pattern generators, it can have a nonlinear impact in the whole system as it moves down. And so instead of having to control every single one of these blue dots, all I've got to do is learn the weights between the red dots and the blue dots and then control the red dots. And I've actually got a much better ability to control, at least in a low dimensional fashion, the movement of the animal. This is all just setting you guys up for some of the stuff we'll talk about in later talks. Yeah, so this is a diagram of what that other thing I mentioned, this reticular formation, and you can think about it in a cartoon form. It's sitting in the middle of your brainstem, and it's coming out and it's hitting these key nuclei in the thalamus, and also spreading out to the whole brain, and it's dousing them in chemicals that sort of bring them into a more excitable regime. We'll talk about this stuff a bit later as well. I'm going to skip over this. This is just like a cool diagram showing you all the different nuclei that are packed into your midbrain and pons, and then all the connections between them. Super complicated, and like we forget about this stuff completely when we think about how the brain works. But there's a lot of complexity baked into these circuits. All right, another really important area that we don't think about enough is the thalamus. So you saw it before, sitting, looking like an ice cream cone sitting on top of an ice cream cone here. It's receiving a lot of inputs from the brainstem, but it also projects out to the whole rest of the cortex. So what I've got here is a picture of the thalamus. This is the anterior portion, the posterior portion, and this is lateral, and this is medial. And the thalamus is broken up into a set of subnuclei, which have different projection patterns. So this red structure here, the mediodorsal nucleus, and I should mention as well, we have two of these, has predominant connectivity with the frontal cortices, whereas something like the pulvinar has connections with the posterior parietal lobe. These little areas like the lateral geniculate nucleus I mentioned before receive all the retinal input and then project it onto primary visual cortex, and then the medial geniculate nucleus is projecting to this primary auditory cortex. Other areas are more involved in motor function, so the ventral anterior and the ventral lateral are really interesting because they receive differential basal ganglia and cerebellar feedback, and that'll become important in a later lecture when we talk about their functional roles. And the anterior nucleus is really fascinating. It projects mainly to this area of cingulate cortex, particularly the posterior cingulate, and it receives input from the mammillary bodies. And this is a really interesting area. There's these tiny little lumps in your diencephalon that if you have a dietary deficiency in thiamine, which you never ever think of, unless you remember back to the old Nutri-Grain commercials. Yeah, Joe's nodding. We're showing our age, Joe. So you don't get a thiamine deficiency so long as you eat food. But if you don't eat food, then you get a thiamine deficiency. And it turns out that a great way to not eat food is to be a chronic alcoholic. So what happens is that chronic alcoholics get a thiamine deficiency by accident because they just drink and they don't eat. They're not getting enough of their vitamins. And so what that does is that completely withers their anterior nucleus, which means that the posterior cingulate's not getting the input it wants to receive. And the effect that has is that your memory becomes completely shot. So people will show up with something called Korsakoff syndrome, which is a thiamine deficiency, which obliterates this anterior nucleus as input to the cortex. So there's lots of interesting things we can learn about that. I just think it's a cool story. All right, another cool thing that happens in the thalamus that we don't talk about a lot, typically people think about the thalamus as really this right-hand side of the figure. So some specific input coming into the cortex and giving some very specific targeted hit, which then maybe gets a knock-on effect to, say, perceive a face or something like that. But it turns out that spread, almost like in a yin and yang fashion, there's an overlap in this complete other population of cells, which we call matrix cells. So we call these core cells and these matrix cells. And what they do instead is they come up something like what the ascending arousal system does and just kind of hit multiple different areas in sort of a semi-random fashion. And this will become relevant to some of the models that I'll talk about in later in the lecture series. But I think we ignore these to our detriment for thinking about how the system works. All right, got about 10 minutes left. I'm sorry if I'm racing through this stuff, but it's good to get a high level. All right, so the basal ganglia, really fascinating set of regions. We'll cover these in more detail later. They're deep to the cerebral cortex. So now what we've done is we've made the cortex see-through. You can see these big blobby regions and these little curly things that run around. The basal ganglia, so now if we sort of like, we take that and then we cut off, so this here, the putamen, if we cut that and we look inside it, the basal ganglia is like this sort of stacked set of nuclei that you can think of as kind of like a set of handbrakes or multi-level handbrakes that are controlling the flow of activity both in the thalamus and in the brainstem. And they're receiving a ton of input from the cortex. So you can think about spread across this realm. There's all these inputs coming in. Anytime the cortex sends out a signal, it's also sending a little corollary discharge off to the basal ganglia. And the basal ganglia, with its little set of loops on top of loops inhibiting one another, can kind of shape whether or not that signal gets a little boost back and says, yeah, this is a good signal to have, or shuts it down and says, this is not a signal we want. So it's really like this kind of like control parameter changing the excitability of the population that signaled it in the first place. We'll get into some of the nitty-gritty of the circuitry. I didn't really want to cover it in this first section, but this, if you look in any kind of neuroanatomy, like clinical neuroanatomy textbook, the circuitry diagram of the basal ganglia is the kind of thing they force poor medical students to memorize for no good reason. The thing that I want to point out in this high level is this idea of a gradient. So what is happening is that every cortical area, more or less, is sending input into the striatum. We think maybe V1 doesn't do this as much, but every other area is sending a little corollary discharge into the striatum. which is then disinhibiting this area called the pallidum or the substantia nigra, which then the idea is then releases the thalamus to do its thing. It turns out that this idea of these closed loops is slightly misguided, and we'll talk about that in some later talks. But I want you to kind of get the idea that the whole cerebral cortex has almost got like this little friend with it that can do certain things that the cortex can't do. So an analogy that I heard someone use once is that if the cortex is the president, then the basal ganglia are like the sort of person in the presidency whispering what to do. You know, oh, I've got to make this decision. Do I bomb this country or that country? And he goes, oh, bomb that country. They suck, right? That's the idea, is the basal ganglia is that little whisperer. So yeah, this is just another way to show you that they're kind of like represented across this hierarchy. And yeah, this is I just put this in because it's really pretty, but it's basically showing you the same thing. Similar, some structures, every structure has got a representative hub in each of these in different parts of the circuit, which theoretically can have slightly different functions. All right, we're getting there. The cerebellum. So I said this was the little brain sitting at the back, but it's got a literal boatload of cells in it. We're not going to cover exactly computationally what this might be doing until the next lecture. But the idea is that the cerebellum is really involved in sharpening signals. So it gets signals from a lot of different places. You can partition the cerebellum up into a few different ways. So imagine now we're looking at the cerebellum from the back. I've just kind of spread it out so that you can see the shape of it. There's areas here in green we call the anterior lobe or the sensory cerebellum, are receiving inputs from the sensory system. Down the bottom here, what's called the flocculonodular lobe or the vestibulocerebellum is receiving inputs from your inner ear, your semicircular canals, which are telling you where you're oriented in space. So as you tip around like this, your cerebellum is getting a little signal that's saying, you just move to the left, you should expect the world to shift with you. So if you've ever seen what's called a vestibular ocular reflex, where people move their head to the side and the eyes go in the opposite direction, that's because you have an intact cerebellum, which is allowing you to calculate how much you moved and then adjust for that. So if that's happening down in the vestibular system, then what the sensory system's doing is it's getting similar kinds of things, but for when I move my arms and my legs around. So let's say that you threw me a ball, I want to put my arm out to catch it. I'll say my plan, my motor cortex is to move my arm in a particular direction. The cerebellum is going to ensure that that happens in as efficient and effective way as possible without me shaking my hand around and then missing the ball. Right? So it's letting you learn the patterns that smooth out and sharpen your movement. The really fascinating areas that I spend my time thinking about what's called the lateral cerebellum, these areas like CRUS 1 and CRUS 2, which have preferentially expanded in primates and non-human and in humans. And instead of feeding back to the motor cortex or the vestibular system, they actually feed back to the frontal cortex. So this area we use for planning, for trying to coordinate our activities. In humans, it's got this really big system underlying it that's trying to sharpen and shape its activity over time. And I think this is incredibly important, and I'll try and convince you of that in the next couple of weeks. This slide is to show you that there's these reciprocal connections. So the same way that the basal ganglia the cortex is sending down to the basal ganglia, hitting the thalamus, and then it's getting the input back. The cerebellum has a similar kind of distributed circuit. So the cortex sends an input down to the pontine nuclei. That's that bulge I told you about that's prominent in humans. It then comes down into the cerebellar cortex, gets filtered around, and some really interesting stuff happens to it. The output of the cerebellum then projects back to the thalamus, which then hits the same area that, well, not always the same, but an area of the cortex, and gives a little nudge or boost. So there's this kind of distributed pattern, both a motor pattern, right, that's going to tell you whether or not to make your movements sharp, but then this cognitive pattern that's going to tell you to make your cognitive system sharp or perhaps more automatic. Yes, and this is just to sort of show you that output coming back up through either the red nucleus or the thalamus, and then supporting parietal, motor, prefrontal, or temporal information processing. Just for any of the gradient weenies in the audience, the same kinds of gradients we see in cortex also exist in the cerebellum. So what I'm showing you here is a flat map of the cerebellum. What I've done here is taken this gnarly kind of, really highly convoluted structure and blown it out like it was a balloon, cut it in the right kinds of places so I can flatten it out. So it's kind of like the those maps of the world, of the globe you've seen, where it's sort of like a bit lumpy in different places. They try not to augment anything, just lay it out flat. And you can see that there's these primary gradients from these structures that are more involved in the kind of cognitive processing down to the more sensory and vestibular processing. And then the second gradient is kind of mapping the lateral sort of structures and spreading them out from this sort of more superior and medial structures. So there's no real take-home message here. It's just to say that it might be that we don't need to partition the brain up into little chunks. It might be that gradients are a better way to explain it. All right, last few slides. This is the hippocampus. So again, instead of just pulling the brain apart like I did with the insula, here I had to actually make a cut. So the hippocampus is actually deep in the brain. It looks a little bit like this if you slice it. So if I was to like perform a section along this axis here, it would have this kind of funny looking shape to it. People said it looks like a seahorse, which is why it's called a hippocampus. I don't know, I don't get it, but anyway. So it has this particular shape. It has different names for different parts. People know the connectivity really, really well. So they know the kind of inputs to different structures and which regions are connected to what. I'm not going to go into those in detail now. I just want you to know about the fact that the hippocampus has got these kind of different areas within it, like the dentate gyrus and these CA1, 2, and 3 regions. But more conceptually, I think one thing that we can take away from today is where the hippocampus sits within this beautiful mess of a hierarchy. So recall here, we've got visual input coming into the thalamus. It hits the primary visual cortex, and it starts to spread out into these different channels. Here, this is going to be the parietal lobe, so what you're going to do with it. And this is going to be the ventral temporal lobe, what you're seeing. So this would be where you're processing whether it's a face or a house or a car. And this would be how do you catch the ball or how do you throw the ball. The hippocampus is sitting right there. So in some ways, it's kind of like the king of the cortex, right? It's actually sitting hierarchically right at the top. And this becomes really important because one of the ways to think about what the hippocampus is doing is it's learning the specific pattern of inputs it gets in a certain context. So let's say the hippocampus is getting fed a picture of my face in the Brain and Mind Center Level 5 lecture theater and talking about anatomy too much that you guys all want to fall asleep. You'll remember that with your hippocampus, that precise constellation of those different details, which you can then reinvigorate as an episodic memory. So I just think that's a better way to, it just makes more sense to me than saying it magically has a memory function. It also makes sense why losing it is really important for things like dementia, right? So if you go and knock that area out, now all of a sudden you're in a context and someone says to you, hey, where'd you leave your hat? And you're like, oh, I don't know. I've got a hat signal and I've got no idea how to coordinate it with room or context or my past experience. So I just think this maybe helps put that into a slightly different framework. The amygdala here, so in this view here, right, we've got our brainstem, our thalamus sitting on the outside is the basal ganglia. As the basal ganglia wrap around, the hippocampus basically sits right nestled next to the ventricles, and the amygdala sits in front of it. I'm not going to talk about the amygdala today, but I just wanted you to get a visualization of where the amygdala sits. It's a term that comes up an awful lot, particularly in clinical neuroscience. And then these are some other areas. It's just to kind of orient you again. That tectum I talked about for shifting your eyes around. Now what we're doing is we're looking at the posterior side of the brainstem. And I've cut the cerebellum off and thrown it away. You can see the pons here at the front, the thalami sitting on top, these little ice cream sitting on an ice cream cone. And the tectum, the colliculi, sitting at the back. And these are the regions that are really important for controlling your saccadic eye movements. I'm looking around into the world. The superior colliculi process visual information, and inferior process auditory. And the pineal gland here is just has some reference because it controls a part of the sleep-wake cycle. All right, last couple of things really quickly. Brain couldn't do anything without blood. So this is the aorta from the heart pumping up into the internal carotid arteries. And there's this sort of diffuse system that sort of basically douses the brain in blood. It wraps around the brainstem in a thing called the circle of Willis, which is really important. As it goes off, it sends off these little branches into all the subcortical structures. So this might be of interest to people doing, white matter hyperintensities or things like that, or MS. There's particular boundaries for where these things go. So these little borderlines are called watersheds. You'll notice that the brain is not given blood in that respects kind of anatomical boundaries. It's more kind of the easiest way to diffuse blood over the whole system. This is the venous system, so this is how you drain the blood back out. There's this sort of little mohawk-looking thing that wraps around the temporal lobes as well. And then there's one that goes around the cingulate. And they're basically taking all of the waste products, the deoxygenated blood, and pumping it back down through the vena cava into the heart. And then the lymphatic system. So this is the central structure for cerebrospinal fluid, which runs all the way down the spinal cord and comes up into these different horns. And basically that's creating this little sort of like saltwater bath that you use to both get some nourishment for cells, but also to clean up waste. So by now you should feel that neuroanatomy is bloody complicated, right? There's so much stuff to it. In fact, someone very clever said it's infinite. I think that's true. What I was trying to do today was just give you a bird's eye view so you can come and now you know what those terms mean in some context. You shouldn't know it all, and you should probably feel overwhelmed because that's a lot. Hopefully what we'll do over the next couple talks is come back to this and dive down into each of the different bits so that you can say, oh, the hippocampus is sitting there in the temporal lobe, and I know that its connectivity means that it can sit on top of the visual hierarchy and it can make contacts and it can reinvigorate those contacts. So I'm hoping that this will drill all the way down in, but you'll have to trust me on that because it's a lot to process. Another really important thing is if you do want to know this stuff, you've really got to do the work yourself. It's one thing to hear someone talk about it. It's another thing to go and look and go make guesses and be wrong and update. And for that, I think that worked examples are the best. So take your disease of interest. I want to understand Parkinson's disease. Where's the basal ganglia? How does that sit in the brain? How is it related to the thalamus, to the cortex, to the arousal system? Use that as a tether. It's the best way in, I think. All right, so if I haven't completely overwhelmed you, the idea is that for the next lecture, which isn't next week, but the week after, we're going to talk about microarchitecture and circuitry. And then in the final lecture, we'll talk about how it all might fit together. And if I forgot your favorite area, anyway, let me know and I can cover it. All right, thank you. And I think there's a recording of this if people want to go back, and I'm happy to share the slides as well.